To improve the understanding of transcription factor DNA binding, we developed a method to measure dissociation constant at large scale using fluorescence anisotropy measurements. This technique allows obtaining automatically full titration curves to determine dissociation constant with high sensitivity. It works in solution at equilibrium, has moderate throughput and large dynamical range.
We applied type of A to refine the binding affinity landscape of transcription factors. However, the protocol could be applied to other kinds of binding events like protein-protein or protein-drug interactions, for example. I advise to first perform multiple titrations with the same binding partners to get an estimation about the reproducibility of the measurements. If available, use an automated system for even better reproducibility.
To anneal the DNA oligomers of the reference DNA, mix 7 microliters of each 10 millimolar dye-labeled forward and unlabeled reverse strand in one tube. For the competitor DNA, mix 20 microliters of each 100 millimolar unlabeled strand into 2 wells of a 96-well PCR plate. Then, use a standard PCR machine to heat up the DNA solutions to 70 degrees Celsius for 3 minutes, and then reduce to room temperature at the rate of 0.1 degrees Celsius per second. Use a microwave oven to melt 0.5 grams agarose in 100 milliliters binding buffer. Add double distilled water to adjust the final volume to compensate.